In a previous video, I tackled the different erythrocyte cytoskeletal proteins. In this video, I will be discussing the first three integral membrane proteins in this list. Rh-associated glycoproteins will be discussed in the next video, along with the ABO blood grouping system. Band3 or AE1 is what is called a multipass protein. This means that it traverses or passes through the lipid bilayer a few times. In the case of band3, 14 times. The image in the next slide will be more informative in this regard. Band3 functions as an anion exchange protein. Essentially, band3 is a channel that allows chloride ion to be exchanged for bicarbonate ion. This is of particular importance, especially with gas exchange, in which erythrocytes play a vital role. At the tissue level, where levels of carbon dioxide are high due to normal metabolic processes, bicarbonate ions that are generated inside the erythrocyte from the hydration of carbon dioxide catalyzed by carbonic anhydrase are exchanged for chloride ions. This exchange is sometimes termed the chloride shift. As blood from peripheral tissues reaches the lungs, the process is reversed. Carbon dioxide is reformed, if you may, from bicarbonate and is exhaled. We will get into these processes in greater detail in the following slides. As promised, here's a schematic model of human erythrocyte band3. Notice how the polypeptide chain crosses the cell membrane 14 times. The N and C terminal regions of the protein extend into the cytoplasm and provide binding sites for several RBC cytoskeletal proteins, as was discussed in the previous video. You can see as well from the list that many enzymes also bind to these band3 polypeptide terminals. The transmembrane domain, that is, the part of the protein that crossed the cell membrane multiple times, forms an anion exchange channel and consists of 13 alpha helical segments embedded in the lipid bilayer and one non-helical segment. As mentioned earlier, band3 mediates the chloride shift or the anion exchange of bicarbonate ions with chloride ions. Because of this chloride shift, RBCs are able to recognize metabolically active tissues and to supply the minimum amount of oxygen to the said tissues. Thus, band3, along with hemoglobin and the enzyme carbonic anhydrase, all synergistically make RBCs the ideal vehicle for oxygen delivery to the tissues. And obviously, the lungs are involved in this process as well, because where else would we get molecular oxygen from but the atmosphere? For that matter, we have to understand two effects which you may have already encountered in physiology, the Bohr effect and the Haldane effect. The Bohr effect refers to the observation that increases in the partial pressure of carbon dioxide in blood, or similarly decreases in blood pH, result in lower affinity of hemoglobin for oxygen. When oxygen dissociates from oxyhemoglobin, deoxyhemoglobin results. Deoxyhemoglobin is said to be a weaker acid than oxyhemoglobin, making it more readily accepting to protons that are liberated by the dissociation of carbonic acid, formed from the catalytic action of carbonic anhydrase. For that matter, most of the carbon dioxide transported in blood is essentially in the form of bicarbonate ion. Now, protonated hemoglobin lowers the affinity of hemoglobin for oxygen, shifting the oxygen-hemoglobin dissociation curve to the right in environments of low pH and high partial pressure of carbon dioxide. Basically, carbon dioxide from peripheral tissues is transported by venous blood to the lungs, where the carbon dioxide eventually gets exhaled. A resting 70 kilogram person produces 200 to 250 milliliters of carbon dioxide per minute via normal metabolic processes. Take note as well that majority of carbon dioxide is transported in blood in the form of bicarbonate, as mentioned earlier. This is where the enzyme carbonic anhydrase becomes instrumental, as what I will show in the next slide. Now, the Bohr effect basically occurs in the peripheral tissues, where there is high metabolic activity and carbon dioxide production. Carbon dioxide from tissues make their way into the plasma, where 5 to 10% of it gets dissolved, with the rest entering the RBCs. Once inside, a small portion of it gets dissolved in the cytoplasm, and about another 5 to 10% bind protonated deoxyhemoglobin to form carbamino compounds, liberating protons in the process. About 80 to 90% of the carbon dioxide reacts with water in the presence of carbonic anhydrase to form carbonic acid. Carbonic acid is quite unstable that it dissociates into bicarbonate and proton shortly after its formation. The protons that dissociate from carbonic acid and the protons that get released upon the formation of carbamino compounds then bind to specific amino acid residues on the globin chains of hemoglobin, and molecular oxygen is thus released and eventually unloaded onto the peripheral tissues. We then have the Haldane effect, which describes the phenomenon by which binding of oxygen to hemoglobin promotes the release of carbon dioxide. In many ways, the Haldane effect is the mirror image of the Bohr effect, making clear that oxygen and carbon dioxide compete for hemoglobin occupancy. This competition is a helpful biochemical feature which facilitates exchange of carbon dioxide for oxygen in the pulmonary and peripheral circulations. The Haldane effect basically takes place in the lungs, where there is an abundance of molecular oxygen relative to carbon dioxide. Oxygenation of protonated deoxyhemoglobin promotes dissociation of protons from it. These protons then contribute to the reformation of carbon dioxide, mainly from bicarbonate. So, in the lungs, oxygen makes its way from the alveoli into blood contained in the alveolar capillaries and eventually into the RBC, where it binds protonated deoxyhemoglobin. At the same time, carbamino compounds get converted back to protonated deoxyhemoglobin and carbon dioxide, with the former contributing to the protonated deoxyhemoglobin pool to which molecular oxygen can bind. Upon binding of oxygen to protonated deoxyhemoglobin, oxyhemoglobin is formed and proton is released. In the presence of carbonic anhydrase, the protons then react with bicarbonate from plasma that re-enters RBC in exchange for chloride ions to form carbonic acid. Carbonic acid in turn dissociates into carbon dioxide and water, a reaction that is likewise catalyzed by carbonic anhydrase. Carbon dioxide then diffuses out of the RBC, and along with the carbon dioxide dissolved in plasma, enters the alveoli, where it is exhaled into the atmosphere. And that ends our discussion on band 3. Next, we have glycophorins. Like band 3, glycophorins are also integral membrane proteins, glycoproteins to be exact, because of the oligosaccharide moieties covalently bound to certain amino acid residues on their extracellular portions. Now, these oligosaccharide moieties are very high in sialic acid content. The extracellular portion is the N-terminal and is hydrophilic. Glycophorins also have an alpha helical membrane spanning portion. The intercellular or cytoplasmic portion, on the other hand, is the C-terminal of the protein. Now, glycophorins are of three basic homologs: GPA, GPB, and GPC. Now, due to the high sialic acid content of the oligosaccharide moieties on the extracellular domain of glycophorins, the overall external charge of RBCs is negative. This is important in preventing their adherence to each other and to the vascular endothelium. Glycophorins also carry a large number of minor blood group antigens. A total of 30 blood group systems are recognized by the International Society of Blood Transfusion. 9 blood group systems, namely ABO, Rhesus or RH, Kell, KID, Duffy, MNS, P, Lewis, and Lutheran, are considered to be clinically significant as these are known to cause hemolytic transfusion reactions and hemolytic diseases of the newborn and the fetus. Glycophorin A acts as a receptor for influenza virus as well, and also for the malaria parasite Plasmodium falciparum. Now, as far as interactions with other RBC membrane proteins are concerned, glycophorin A interacts with band 3 as part of a macromolecular complex. Glycophorin C, on the other hand, associates with protein 4.1 and the P55 scaffolding protein, thus providing an additional contact site between the cell membrane and the cytoskeleton, thereby stabilizing the RBC membrane. This fact was touched on in the previous video. The third important integral membrane protein to discuss is glucose transporter 1, also known as GLUT1 or glucose permease. As I mentioned in an earlier slide, GLUT1 internalizes glucose from the plasma into the RBC via facilitated diffusion. GLUT1 is vital to the RBC because it needs glucose to generate ATP, since RBC can only do that via glycolysis. Since their mitochondria are removed during the course of their development, mature RBCs cannot rely on the Krebs cycle and the electron transport chain for their energy needs. Flashed here in this slide is the entire glycolytic pathway, which you will eventually learn in the second semester under glucose metabolism. There is an alternative pathway, however, in mature RBCs that branches right after the step catalyzed by glyceraldehyde 3-phosphate dehydrogenase. In this alternative pathway, 1,3-bisphosphoglycerate is converted to 2,3-bisphosphoglycerate, or 2,3-BPG for short, via the action of the enzyme bisphosphoglycerate mutase. We call this pathway the Rapoport-Lubering pathway or cycle. 2,3-BPG, if you could still recall from earlier lectures, binds to and stabilizes hemoglobin in its taut or T state. This molecule likewise regulates oxygen release from hemoglobin and delivery to tissues. Now, the concentration of 2,3-BPG varies proportionately with pH, meaning a decrease in pH within the physiological range decreases the activity of the enzyme bisphosphoglycerate mutase, which catalyzes the conversion of 1,3-bisphosphoglycerate to 2,3-BPG. This ensures that 2,3-BPG levels rise and fall at appropriate times during the oxygen transport cycle. Let us now discuss select clinical conditions that involve abnormalities in integral membrane proteins. Southeast Asian ovalocytosis, or SAO for short, is characterized by specific RBC morphology, often described as stomatocytic erythrocytosis. This means that peripheral blood smears show large oval red cells with a transverse ridge across the central area, hence the term stomatocyte, pertaining to the mouth-like or slit-like pattern that replaces the normal central zone of pallor. Now, this disorder is largely asymptomatic and is caused by a defect in band 3. SAO is seen in parts of Southeast Asia, including Malaysia, New Guinea, Indonesia, and the Philippines. In the Melanesian population of Papua New Guinea, as many as 12 to 30% of individuals may be affected, which is why SAO is sometimes referred to as Melanesian ovalocytosis. Hereditary stomatocytosis, or HST, is a rare inherited disorder associated with abnormal membrane permeability and red cell cation content. Mutations affecting band 3 have been reported in HST. Recall that one of the functions of band 3 is as an anion exchange protein that transports bicarbonate ions out of the RBC in exchange for chloride ions. The bicarbonate ions are used to transport carbon dioxide produced from tissue respiration. HST-associated mutations in the gene encoding band 3 are believed to turn the anion exchanger into an unregulated cation channel. Now, this abnormality in ion exchanger function then results in either overhydration or dehydration of the RBCs. Recall as well that band 3 also contributes to the structure of the RBC cytoskeleton by linking ankyrin to the RBC membrane. Abnormalities in band 3 can also lead to RBCs having the shape of a uniconcave cup instead of having the normal biconcave shape. This results in RBCs acquiring a mouth-like or slit-like pattern that replaces the normal central zone of pallor, as you can see in this image. Lastly, we have acanthocytosis. Acanthocytosis is a red blood cell phenotype that is associated with various underlying conditions. Acanthocytes are spiculated red cells with a few projections of varying sizes and surface distribution, as seen on peripheral smear. While the genetic forms of acanthocytosis, particularly the ones involving abnormalities in integral membrane protein and cytoskeleton protein structure, or with inherited movement disorders, are rare, the much more common types are those that are secondary to relatively more common underlying clinical conditions, such as liver cirrhosis. Here is how acanthocytes look like on peripheral blood smear. Now I will end part 3 of this video series here. Please proceed to part 4, where we will discuss the Rh-associated glycoproteins and the ABO blood typing system.